In the first half of this session, I will talk about 3D mapping of activity of grids and Stereo-EEG. And in the second half of this slot, my colleague Mostafa will tell you about his research in high-frequency oscillations. So when working with intracranial EEG, you usually acquire many channels. So it can be 100 or even more. And therefore, you get a lot of data from different locations. And when you do an ECOG or a Stereo-EEG experiment, you usually want to define the corresponding montage, which represents the location of these implanted electrodes, because this is required for the topographical visualization. If you want to be able to do a easy result interpretation, for example, by showing activation in terms of bubbles, or to show activation with a heat map. Or if you want to do some advanced analysis, for example, based on anatomical regions. So you are only interested in certain electrodes which were inserted in a certain region, and only do the analysis based on these electrodes. And you also need to know the locations of your electrodes if you want to compare your intracranial EEG data to other modalities, for example, fMRI and DTI. And for this, you also have to know the 3D positions, because otherwise you can't compare it to this volume-based results. So the easiest approach to define a montage is just to use a schematic image. And this is the way how it's done in the old cortical version. So you just have a schematic of the brain, and you see the channels arranged. For example, here, one grid and one depth electrode. And it's nice because you know the channel numbering, you know which channels belong to which grid, and you can estimate where the grid is located. But you can imagine that, especially with depth electrodes, it can get quite confusing to put all of them on one schematic image. So another approach is to use a photo, for example, of the open skull, and just overlay the electrodes on that. So for example, when you do a wake surgery, you don't have, in many cases, other images available, just a photo. So you have to do that. Later on, I will show how to get a 3D montage if you only have a photo available. And for depth electrodes, another approach is, for example, to use MRI slices and use that to visualize the contacts in 2D. That's a little bit easier to see than just with the schematic image. But usually the goal is to work with 3D. And what you can always do is to utilize a template brain. So a very commonly used one is the MNI template brain. And you can visualize your electrodes on the template, and you gain all the new features which are available in 3D. So you can rotate and so on, and see especially depth electrodes very nicely. But the best, of course, is to really create a subject-specific brain, which really corresponds to your subject. And for this, you need a T1-weighted MRI. And in order to locate the electrode positions correctly, you need a postoperative CT. And all of these different possibilities can be covered by the EEG Montage creator. So depending on the input data you have available, you can create the corresponding montage. And the output is always a single MON file, which you can then use for real-time mapping in GHiSys or for offline analysis in GBS Analyze or your own MATLAB script. So the application has 4 importers, which correspond exactly to the cases I explained before. So we have the default one, which is a 3D montage based on the MNI template brain, or you can start the montage based on an image or FreeSurfer or any other files. So usually we go with FreeSurfer, and you already saw some nice presentations with nice 3D renderings of the brain. And I think that nearly 95% of all these renderings are based on FreeSurfer. So let me explain a little bit what FreeSurfer is. So FreeSurfer is a free tool which you can use on Ubuntu and Mac. And it's basically just a console application where you input the T1 weighted MRI as argument, and then the processing stream runs for a couple of hours, and it produces many files and folders. And for example, it produces this very nice and accurate 3D model of the cortex, and it also provides pastillation information. So it labels all the vertices so that you get a labeling of the gyri and so on. So it's a really powerful tool. Like I said, FreeSurfer can be used only on Ubuntu and Mac and so on. But because of the Windows subsystem for Linux, the EEG Montage creator, which is running on Microsoft on Windows, can utilize FreeSurfer directly. So you can install it through this Windows application. And then you can import existing data, or you can input your MRI input and run FreeSurfer and create all these files directly from the Montage creator. So the fourth importer is the other importer, which allows you, on the one hand, to create a cortex based on any other file, for example, a gray matter segmentation created by SPM. But you see that the surface is not that accurate and nice like in FreeSurfer, but it's way faster. So you have this option here to create the montage based on such data. And what you can also do with this other importer is to add any other files to your montage. So for example, you can add DTI tracks as custom surfaces. So let's have a look how this is done. So here we are loading an existing FreeSurfer result. So you see that the application loads the MRI, it loads the pile file and so on. This is the cortex segmentation. Then it loads the annotation, so the coloring of the cortex and some other files. Then we can switch to the other importer, and now we want to add some DTI fibers, so DTI tracks. So we can load the file. This file was created by 3D Slicer. We can set the color and give it a meaningful name. And here you see, or you can already see the fiber tracts. And now we see the MRI, which the 3D brain is based on. So this is all provided by FreeSurfer and already available in your Montage. And we can even create subcortical regions, in that case, the 5 segments of the corpus callosum, because we want to add that to our 3D Montage. So we can also visualize this segmentation volume, which is created by FreeSurfer, and use that, for example, to clip the cortex model to get a very nice view inside the brain. So you see, just by importing the FreeSurfer data, we already have a lot of anatomical data available. And now our goal is to define the corresponding electrode locations with respect to the imported data. And the Montage rate is structured as follows. We have some workflows for each modality, so for ECOG and for Stereo-EEG, and you just go through all the processing steps in order to determine the electrode positions. So now I will show some examples and some procedures. We use these procedures for different studies, and it just depends what you have available and what you want. So in the first example is to interactively place an ECOG grid and a Stereo-EEG shaft in 3D without any available CT data. So we're using the data we've imported beforehand. And now we started with the ECOG workflow to place electrode grids. So the first step is to compute a hull which covers the cortex. And yeah, you see it's a tight hull. And we need that for the interactive electrode grid placement. So in here, we can show the pacillation information, because we want to place a grid over the motor cortex. So now we can add a grid. We can select it from a big grid library with all the manufacturers. And now we can interactively place the grid over the region we are interested in. So here, for example, over the motor area. And in the next step, we want to add a depth electrode. And we switch the workflow. And just now we can first hide all the stuff we don't need at the moment. So we hide the DTI information. And now we can also, in this step, temporarily create a subcortical structure of the thalamus, because we want to place an electrode shaft in the thalamus. So we can select the corresponding electrode, and then we can interactively move the entry and the target point like we want. And place it. We can also view it from multiple perspectives, which is very important when working with Stereo-EEG electrodes. So now we added the depth electrode, and then we can save it to our Montage. Of course, usually you add many grids and many depth electrodes. So this you can do if you don't have a CT available. So you can do the positioning based on anatomical structures. So the next case I want to show you is how to do interactively echo grid placement with an overlay image. So that you need, or you can use that if you have a awake surgery case where the electrodes are only inside the head during the surgery. So usually you don't have a CT available. So, but you can take an image of the open skull and you can overlay that image on the 3D brain, and then you can adapt the view angle and the zoom of the camera to match the perspective of the camera you used when taking the photo. And then you can overlay and place the grid with the help of the photo. So you can scale it and move it to match the photo. And like you see, you get a pretty good and accurate result. And this is really nice because without TT, it's normally really hard to create a 3D model. So the next case I want to show you is how to do depth electrode placement if you have a CT available. So let's have a look at this. So again, for the subject, we already have some free surfer data available. So we import all the files into the Montage creator. And then we select the corresponding workflow. And again, then we need to compute a hull beforehand. This is required for the placement afterwards. And in the next step, we can load the CT. And the CT and the MRI, the 3D model is based on, are not aligned by default. So we have to do a registration. So here the algorithm automatically registers both image modalities. So green is the CT and the pink one is the MRI. And you see it really matches now nicely. And in the next step, you can segment the CT and get rid of all cable artifacts and so on. So everything outside the brain is cable, and we can remove that in that step so that we only get the contacts of the electrode shafts. And now we want to get the actual depth electrodes. And the nice thing with this model is that it automatically determines the electrodes and place them based on the trajectories, which are automatically fitted into your segmented CT. And of course, you can change to the correct electrodes which were used. And yeah, this is pretty nice because when you have many depth electrodes, this approach is really fast. Of course, you can change, remove, and add shafts manually, like you see here. So again, from the database, you can search for your electrode and add it. And then you can, again, move the entry and target point. And you can snap it just to the trajectory and it will automatically fit. So you can snap to the trajectory, or you can also snap contacts to the CT, to the segmented CT blobs, the white ones you see here. And you see that some of the shafts are marked red. That means that some of the contacts are outside of the of the brain, and you can exclude them from your montage. This is also possible in that module. And then you are done within a few minutes, and you can save your montage and do the actual analysis. So the next thing I want to show is the same for ECOG electrodes. So again, we do the registration. So you see that the CT and the MRI are nicely registered. And then again, we do the segmentation, but now just for the ECOG grids. So you see the electrode contacts within the grid. And in this step, we can automatically detect the electrode positions. And in the next step, we can correct that a little bit if something was not detected nicely. You can add electrodes, remove them, and move them around and so on. And now you see that some of the electrodes are within the brain a little bit, and that's because of the brain shift between preoperative and postoperative data. And in this module, you can compensate for that. So we project to this brain hull we computed beforehand. And in the last step, we can actually assign the grid shapes and restore the actual contact spacing. Because of the projection and so on, there might be, the grid spacing might be changed a little bit. So we can here assign from the database the grid to our determined electrode positions. And then it will create a third type of position, which are corrected positions. And you also see the grid shape. Yeah, the last example I want to show is how to do depth electrode placement if you have coordinates available. So for example, from older experiments where you used other tools, or you get coordinates from a neural navigation system, you can use that workflow to create the 3D montage. So here you can load a CSV file with the positions, and you can add them to your model. And again, in this electrode assignment step, you can also assign depth electrodes. So from the list of contacts, you select, for example, in 7 contacts which belong to a shaft. And then from the dialog, you select the correct electrode device, in that case, that one. And you see that one contact was excluded, so we can also exclude that here. And we just have to assign the first and the last contact, and it will automatically assign the whole depth electrodes. So this is also a very fast way to assign the electrode shafts when you have electrode positions and locations available from any other tool. Yeah, and what you can do now with this montage, so you can use it, for example, in GHISIS Simulink. So we have a couple of 3D scopes available. So a bubble scope to show activity with bubbles, or an interpolation scope to show activity interpolated on the head with a heat map. And it's a demo. So we can load this montage and show activity in real time. And of course, all these visualizations I showed before with the DTI tracks and everything, you can control even in this 3D scopes in our GHISIS environment. So also in this scope, during the visualization in real time, you can create the subcortical structures and so on to yeah, in real time show everything and analyze it. Of course, you can also import the file, the montage you created with the Montage creator into MATLAB. The good thing is that everything, all the data is bundled into one file, so you can load it, and then you get a big data structure, which you can then use for visualization purposes. So we use that, for example, in a study with Dr. Sanada, where we visualized ECOG result, ECS results, and fMRI results, and then we compared these mapping modalities. And for this, it was crucial to create a nice 3D montage for all patients to do a good analysis. Yeah, and of course, with MATLAB, you can then create very high resolution renderings for your publication. So you can use the montage for real-time stuff in Quad-TQ, or in HYSYS, or you can use it offline in MATLAB, for example, for your publications. Yeah, thank you very much. And now my colleague Mostafa will tell you something about his high-frequency oscillation research.
Maybe MJ, a quick question before. Do you know why it's called Stereo-EEG? Does it has anything to do with stereo sound left and right channels? Do you know?
I don't think so.
Mustafa, do you know? Why is it called Stereo-EEG? Do you know? You have to log in, otherwise we don't hear you. So maybe I can answer it. So Stereo-EEG is not coming from the stereo sound stuff with left and right ear. It's coming from stereotactic EEG, because you need a stereotactic device to insert a kind of needle electrode into the brain. So you need a 3D environment to know where the electrode is going. And even with such a stereotactic device, which is guiding the electrode, sometimes the electrodes are ending up in a different brain region. So Dean Grosienski had such an experiment where the Stereo-EEG reached the hippocampus, and then they did a P300 spelling experiments with data coming from the hippocampus. This was a very nice publication, and nobody expected that the hippocampus has a P300 component for spelling. But with an accident, this was actually positively proven. Now we are moving to Dr. Mostafa. So he just received his PhD a few days ago. How was your defense? Difficult, easy?
Not so difficult, yeah.
Not so
happy.
difficult. So he learned a lot, which is good. And Mostafa spent a lot of time doing his PhD about high-frequency oscillations. And they are very nice because they are considered as a biomarker for seizure onset zones. There are very difficult terms around this topic, so Mostafa is going to explain you all of them and how to detect HFOs. Please, Mostafa, nice to have you. Go ahead.
Thank you, Christoph, for having me. Okay. The topic is high-frequency oscillation and epileptiform. This kind of the epilepsy biomarkers that we want to review today. So for the epilepsy biomarker, in the pre-surgical evaluation or in the surgery planning, two things should be done. First, some area or pathological area should be found from one hand, and the other hand, the functional part of the brain should be preserved. These two things that usually neurosurgeon do. Today, I want to recap the pathological part or the epilepsy biomarkers, and in the upcoming days, my colleagues will talk about the functional area of the brain. So for the EEG biomarkers, we have several biomarkers that the gold standard actually is the seizure onset zone. This zone can be found in the epilepsy monitoring unit or EMU by monitoring the patient for a week or 10 days like that. And next to it, we have different biomarkers. We call it a spike, or some people call it the epileptiform discharge. Then we have a high frequency oscillation. These two biomarkers are well known in the epilepsy society. The next biomarker is epileptogenic zone. Actually, this zone is theoretical zone, and we can find it by the overlapping the existing biomarkers. And for finding how good is this zone, we usually compare with the seizure outcome. If we have a good seizure outcome, then we can say we finally managed to find this epileptogenic zone. In the right image, you will see different area that can be detected by the EEG recording or ECG recording. Here we have a seizure and we have a IED, or IED actually is interelectrical epileptiform discharge, or we can call it a spike. And we have a HFO. As you can see here, we have the overlap between these markers. Okay, then at the end, our main the target is the epileptogenic zone that can be found by overlapping this area. So let's start with the most well-known zone here. In this video, you will see how we can monitor the patient in the EMU. For example, you will wait for example, 10 days or a week. In channel number 105, you will see that the seizure initiated. For example, this area can be noted or can be considered as the seizure onset zone. This area is the interest for neurosurgeon for doing the resection. As you can see in the right panel, we can see it early in the high frequency domain than the low frequency. Then it would be the propagate all over the brain. According to the neurosurgery routine, they should have found 3 times or 3 seizure to confirm for the surgery. So the next biomarkers here, we have a high frequency oscillation. Is any neural activity with the limited duration and the frequency component. Any HFO has the following parameters. For example, it should be between 4 to 6 cycle, and the amplitude should be between 10 and 100. And the average of the HFO should be greater than the surrounding area, and the duration should be 6 milliseconds. And if you have some consecutive the HFO, the distance between two of consecutive HFO should be 10 milliseconds. This is known as the high frequency that can be detected in 2 different range between the 80 to 250. We call it ripple, and 250 to 500. We can call it the fast ripple. In the right side, you will see the epileptiform discharge. As I said, it has a different term. Sometimes we call it a spike and interelectrical epileptiform discharge and different things. Here, you will see some spike if the sharp activity is between 2 to 70, we call it a spike. If it is between 70 to 100, we call it sharp wave. If we have some the repetitive spike, we can call it polyspike. If that sharp area is greater than 200, then we cannot consider as any epileptiform activity. So the HFO itself is divided into two parts. We can call it the pathological HFO is any unprovoked the HFO that carried by any spike activity. In the left image, you will see any spike. Okay, on top of that, in different frequency range. In the above trace is the wideband EEG or ECG. In the below trace is in the high frequency range, for example, greater than 80 hertz. Here we can see this oscillation is carrying by the spike. That's why we can call it the pathological, the HFO, because a spike itself is the pathological activity that carries this sharp activity. On the other side, we have the physiological HFO or physiological high frequency activity. Is any activity that evoked by any task. For example, in right image, you will see the a corticue paradigm that provoke a task, and we can see the high frequency activity. Or in some case, we can see some continuous waveform. In this case, also is the physiological activity. For the pathological, we usually have a short period of the activity. So here we review some epilepsy biomarkers that we want to use it. For example, for the seizure onset zone, we need to have, for example, a week to find it. In some case, usually neurosurgeon cut the drugs or to provoke the seizure, or in some case they put a flashlight to provoke a seizure. That is a little bit suffering for patient. So here we want to show that we can find those seizure onset by the existing biomarkers here. The good point is that we can speed up the monitoring. As I said, for a week or 10 days is a lot for the patient, even if they are kids. That's why by using short period of time, we're trying to show that we can find the same location. So at the end, we don't need to wait for seizure to confirm the seizure onset location. So here, as we see in the previous slide, we had a seizure onset location. As we reviewed the data, we found that we have different seizure onset zone pattern. That's why it was a little bit difficult for the neurosurgeon to find those patterns by eye. That's why we found it's better to quantify those areas, not subjectively. Here, when we categorize this data, we find we have a 6 pattern of the onset. Here, the idea is to classify this seizure onset zone pattern. Here is our pipeline that we managed to classify those areas. First, we had the raw EEG data. We apply some pre-processing like the high-pass filter, comma average. We mark the signal with a 3-second length after each seizure onset location. Finally, we had a pull of the onset patterns. We started to extract feature in 3 domains: time, frequency, and time frequency domain. As you can see here, we have a bunch of the feature. Then at the end, we use the LDA to classify the onset pattern to 6 class of the onset and 1 to non-seizure. So here in the frequency domain, we see that we get some onset can be differentiated in the frequency, even though we have lots of the feature in different domains. Before applying to the LDA, we need the data to be normalized and log transform. Here you can see how data is transformed to have a standard deviation of 1. Then here is our result for the classification. Here is the individual class. You can see here is the overall metric of the classification. Here we managed to classify 6 of the onset with the accuracy of the around 80%. So up until now, we managed to find the seizure onset soon, and actually we managed to quantify the seizure location. Then we move to the next biomarkers. Our next biomarker is the spike, or it has different name, IED. Here is the feature for the spike. It has a sharp wave and a positive baseline shift and a slow wave. As you can see, before a seizure happens, we can see that this spike is happening. That's why we try to find the seizure onset zone by the spike. A spike itself has a different shape, and in some case could be artifacts. Here, the idea is to classify the true spike, or if we want to filter the true spike shape. In this image, we see we have 2 different spike or epilepsy form. In the left panel, actually, is the true shape. We see that is the asymmetrical. It's usually followed by a slow wave, and it's a triphasic, and there is a different duration from the background rate, and the background is irregular. On the other side, you'll see that actually is the artifact. We see that the background is normal, and we see the symmetrical, and we don't have a slow wave. So here we want to use the clustering algorithm to classify between the true and the false spike. Here we use the existing the detector. A whole detector in the literature usually work based on the sharp activity. They usually have energy function. Here we use the signal envelope as an energy function. Here first we detect the whole spike, and by the clustering algorithm we try to separate the true and false spike. The clustering algorithm we use the basic clustering algorithm. The name is the Cummings. Cummings core function actually distance function is Euclidean distance. Here in this image we see how Euclidean distance compare the sample by sample of two spike. Actually, in the reality, the spikes are not fully aligned to each other. So we need another metric to compare these 2 wave shape better than the Euclidean distance. Here we use the dynamic time wrapping. Actually, is kind of the optimization algorithm. It's try to find the closest sample to the between 2 samples. Here we see these metrics, and it compares sample by sample, and at the end it finds the optimal path. When then we replace the core the distance metric of the Cummings by the dynamic time wrapping to find the best comparison. So here we see the clustering algorithm. At the end, we didn't want to have a fully automated algorithm. We actually implement in the semi-automatic manner. Actually, end user choose the cluster who that mimic the true shape of the spike. Here in the red marks, we see that these spikes are similar to the true one, but these below one are usually flat. We cannot consider as a true spike. In the below image, we see that detected a spike and the non-spike, and we see actually they are in the seizure onset zone in the 2D brain. Here, with the 12 patient we had, we managed to find 37.21% of the spike exactly located in the seizure onset zone. In the below image, we see the two groups are different based on this distribution. In the literature, they usually use the biomarker rate to compare this to algorithm. Here we add another metric to compare how good was the detector. Here we use the distance metric, actually measure how close those markers are to this epicenter of the seizure. Here we see for the true spike, or we can call it IED, is much close to the epicenter, is around 2 centimeters. But for the other group that were not the true spike, is very far to the epicenter. So for the next work, we use the data from the Mugomino Hospital with collaboration with Dr. Kamada. We had 3 cases, and we tried to evaluate our implemented biomarkers. So in the inferior part of the brain, we see the seizure onset zone, and we try to find the overlap between our markers and the seizure onset zone, and we want to show how close are they to epicenter. So here we see one example of the detected spikes or epileptiform discharge and the high frequency oscillation. And in this sample, we see that they are located in the seizure onset zone. In this experiment, we had 2 types of the data. We had with the same patient, with using the drug and not using the drug. Here is the below row is the case that didn't use the drug. Actually, a doctor used this drug to diminish the seizure or the rate when they are in the hospital. As we can see, for the patient who didn't use the drug, the rate are higher. Here we want to show that in any case we can detect the markers. Here we introduce the markers. This is the existing markers, the spike and HFO. HFO is also divided into category, ripple and fast ripple. Here is our introduced marker that we call it pathological HFO. Actually, we combine the spike and HFO. This is another type of the spike that actually come in the bunch of the spike that happen next to each other. Even though we see some rate are low here, but the location are focal to epicenter. But in the spike, we can see is spread all the brain. In the next metric we use, we compare our the markers to the epicenter of the seizure. Here we in 2 experiments using the drug and not using the drug. Here we see that our markers is much close to the epicenter than the other biomarkers in our proposed markers that are close. So these were our biomarkers. At the end, we want to establish our works to the hospital. Up until now, we use we process the data offline, and for example, half an hour or 1 hour. Here we want to establish our data to using operating room. So here is our proposed method. Our proposed method actually actually is kind of the adaptive thresholding. When we move through the signals, it adaptively changes the threshold to find the markers. Here is our simulating model that first record the data and do some processing, high pass notch, and pass the signals to our core detector. Actually, here we used half a second of the data to process. In the right image, we see our pipeline. Here we have the signals after filtering and calculating the energy function. We need a threshold to detect the spike. We use for the threshold, we need the mode and median that is detected by the maximum likelihood estimation. Actually, it estimates the parameter of this energy function. When after a short period of time, we could reach the stable detectors, and we see in the next slide. So here we applied our model into 2 patients, and we can find the spikes, and we can also plot it in to the brain. We also compare our results with the existing the detector that were offline. In the above image, we can see after minute 3, the results are stable and it can detect the spike. Here, you can see the accuracy of the model and the F1 score. According to the F1 score is the superior than the other method. F score is comparing the model output that is the balance that try to balance between the 2 groups. The reason is that is low because the number of the non-spike is much higher than the spike. In the literature, usually when we want to compare to algorithm, we count the number of the detected. But here we work on the time sample and we try to compare sample by sample. For example, first we had a data set, we mark the data and we compare the data set with the detected output. That is why, because we are comparing on this time, the number of the the spike is much higher than the spike. That's why we have a lower number of F1. So here we can see our detector that work in online. After a while, then the bubbles pop up and we see which area is important. Then when we move to the times and it will change. Then we don't need to have for example, half an hour data or 1 hour data to calculate, and that can be operated in the real time. So, okay, that is it from my side, and thank you for attention.
Thank you very much for the nice presentation. So Nick is asking, can you really detect HFOs with EEG recordings?
Yes, can be recorded, but it's a little bit difficult because we have many noise when we have a EEG from the scalp. But with the pangolin and with the EEG, we managed to find, but the rate is not so high. When we use the ECG, it's clean data that we have many HFO, but for the scalp EEG, we can have, but the rate is very low.
So when you're using the scalp EEG or the pangolin, how do you find the regions where you expect the HFOs?
Usually we find in the temporal area when we analyze data, temporal and frontal area, we manage to find some HFO. But we have to notice that we have to reduce the frequency, because in the ECG data, we usually calculate between greater than 80 hertz. For EEG, we usually work on the 60 hertz, 60 to 250.
So how long would be a recording that you find HFOs with ECOG compared to EEG recordings?
For ECOG, we can find any HFO within 10 minutes. Usually in the literature, they use it. But for EEG, that we got the data from the Dr. Kamada, we managed to find within the half an hour data.
Do you find HFOs also in healthy people, or is this exclusively for epilepsy?
As I said, HFO, pathological HFO is a little bit difficult to find because the period is very short. But for the healthy patient, we can find, but it is a continuous HFO. We call it physiological HFO because it's related to a task or when patient doing the task or subject doing task, we can find.
And how would you say does the electrode size and type matter? You know, the HFOs are very spatially localized. So if the electrode becomes bigger or smaller, so how is the influence of this size?
Actually, I'm not sure about the size, but for the grid we have here in the g.tec, we managed to find, but if we use the tiny, it's a little bit difficult because of the impedance.
So what's the standard size that you're using when you're doing ECOG recordings or EEG?
Actually, I'm not sure about the size, but we use the data in our system in the IEG montage creator. I don't remember the size, but we use that montage.
So standard clinical grids have opening diameter of about 4 millimeters, and the center to center distance is about 1 centimeter, so that you get an idea. So what would you expect with high resolution grids where the diameter is smaller and the distance is smaller?
If the distance is smaller, we can find, but we have to be sure about the impedance. If the contact is good, we can find any epileptic activity or sharp activity.
Okay, so thank you very much, Mustafa, for the nice presentation.